Okay, so good evening, everybody. We are in the week 9th, and we are going to discuss uh, in terms or in requirements of your syllabus, that is basically the structure of I and some basic functions regarding its structural configurations, that how it is related and what aspect that is basically responsible for these, or apart from these we are also going to discuss about the diseases that basically occur due to the impairments of the vision. Okay, so let's start with the first slide. Okay guys, so see, you can easily see the structure. This is called your eye, right? Now before going to discuss about the structure, like we are going to discuss about what we are going to basically discuss about today is we are going to understand the eyes from the basic concepts, individual structures, detailed structures, and the importance, why it is important in terms of your function. Okay, so if you can see the screen you can find there are different structures, right? Cornea, iris, pupil, it is mentioned, anterior chambers, optic nerve, retinal arteries, veins, macula, choroid, etc., right? Now let me make you much aware about the structure in three dimensional. Okay, so let's go to the next link. Okay, just a minute, I have already opened your link here. See, now if you can see the structure here, so see, this is your eyes. Okay, now this structure, let me a bit zoom. Okay, now see, understanding eyes in two-dimensional and in three-dimensional is very tricky. Now why I have opened this figure is to make you aware about the three-dimensional architecture. Suppose let your eyes is like this, right? Just imagine that we have, what we have done, we have take a eye and keep in front of the screen, right? Now we need to understand the structure, right? So if you can see here in white color, just let me move in this way. If you can see in this position, the outermost layer. Okay, the outermost layer. Okay, the outermost layer in this figure, if I will just like to draw here, if you just pardon me for the timing, if you can see this layer, okay, this layer, this is basically the outer covering. Okay, this outer covering is called your sclera or sclera. Okay, and if you can see here, this portion, right, this portion is your cornea, right? So these two is your basic covering. Okay, if you see here, just a minute, if we see here the structure, this outer layer, if my cursor is visible, this outer layer is basically sclera, and this area is cornea. If you see three-dimensionally, then you will find, if you see two-dimensional, you will find this is an outer layer. But if you can see in the three-dimensional structure, then see, this cornea is basically a it is basically a opaque structure. You can see like this, or you can see like this. This is basically a converging film like structure. Okay, so these two structure is basically protecting your eyes. So the external covering of our eyes is basically made up of sclera and cornea, right? Now let me introduce you guys to the structure inside it. Okay, what is this? It is mentioned choroid, right? So sclera is your outer protection. If you can see here, the sclera is your outer protection, right? Cornea is the outer protection in the frontal plane. It is basically a conversion. Okay, now these two, okay, these two is what these two are meant to protect. Okay, we talk about that the sclera and cornea is basically your protecting structure, right? Now if they are protecting structure, now what they are supposed to protecting? Okay, Now the structure that is they are protecting is your choroid. Okay, now what is choroid? Choroid is the layer between the sclera and retina. Okay, now let's discuss only about choroid. So choroid is the most nutritive area. It basically comprises of lots of blood vessels, lot of muscular layer, lot of nutritional glands, and not nutritional structure, rather nutritional supplies. It has a lots of nutritional supplies, and this region is basically mainly comprises of oxygen and blood vessels. Okay, now let me make you more clear in terms. See, we have a structure that is protecting. What it is protecting to? Numbers of blood vessels. Now why all these things are there? To protect, to provide nutrition, to support the structure that is called your retina. Okay, so the principal structure that is responsible for our vision is your retina, and this is present, this is mentioned here. It is also mentioned retina, you can see in your screen, right? So it is the retina that is responsible for all the visionary action. Okay, this is what the structure of eyes, right? Now if you see the front part, you can see like this. So you can see here in blue, you can see something, right? Or this is here also, you can see in brown. So this is basically your iris. Okay, so iris is nothing but it is associated to the ciliary muscle that is present here. You can see here just after choroid. Okay, and this iris is basically a structure that is responsible for entrance of light. We'll discuss in later. Okay, another more structure is the pupil. Pupil is nothing but a window. Okay, that is responsible for the convergence of light. Okay, now you can see one more structure here. If I'll just show you in 3 dimension, you can see here in white, this is lens. Okay, after the pupil you can find the lens. So lens is very critical to converge or diverge the light and maintain a focal plane or form an image in the retina. Okay, so the image formation occurs in retina. Now retina has a very limited property, very specific property, that it has two type of cells. Okay, one is rod and one is cones, and that is basically responsible for the vision, that is basically responsible for vision in the light or dark. Okay, and we'll discuss in later in this slide, in upcoming slides only, that what basically the vision is. Okay, so hopefully you guys have a bit clear concept about the 3-dimensional structure of eyes right now. Okay, now once if it is clear, you can see the nerve, right? So retina is connected to the optical nerves, that's what I can say. Okay, that is basically a part of choroid plexus. Now let me discuss this point. Okay, so what this is basically meant for: if there is any light is coming from here, then the light transfer to, this is your iris, you can see, and after iris there is, see, I told right, when light is coming, it is first facing your cornea. After cornea you will find your iris, and after iris you will find your pupil, right? And after the pupil, the light is focused in the lens. Okay, and the lens is going to focus your light or at a particular point in the retina, and that is basically results in the image formations. Okay, so we are not going into the details, rather you should clear about the concept. If you are not clear, that just let me repeat that. You can see here in blue, just let me make this right. When light is passing here, passing through here, this is your cornea, right? This is your cornea. Now this light passes through your iris, and further iris it is passes through your pupil. This is your pupil, right? This is your pupil. Then further light passes to your lens. Okay, and this lens is basically responsible to converge this to the, here it is suppose your retina, right? Retina. So it is the retina where the image formation takes place, and that basically visualized by us. Okay, iris have different colors, it is mentioned eye color, and basically it is the iris that determine which color of eyes you have. You can, it can be brown eyes, it can be blue eyes, right? Okay, so now comes to the first question, that what diverges light rays. Okay, now there are different things we can find, right? Just let me discuss very briefly that, see, this is a image. Okay, here it is your image, right? Now if this is a convex lens, so basically convex lens is like this. If you can see here, the convex lens is like this, and if you'll find convex lens produce your virtual image and erect image. The way it is, you can find it like this, but this is large image. Okay, so convex lens is basically results in the formation of large image, that is virtual image. However, concave lens is basically for help in the formation of real image. Okay, now if you can see, suppose here is the image. Okay, so suppose here is your image, and if this light passes like this, so this is converging, right? This is converging, right? And here it your object, image is, object is formation, sorry, image is forming, right? So this is your converging. So convex lens is convergence, converging, or sorry, convex lens is, okay, just a minute, convex lens is converging, right? Convex lens is converging, that's why it is convex, right? Converging. But concave lens, what it is doing, it is diverging. It is like this. If it is like this, okay, when it is not forming, it is forming real image, and real image is always inverted. Okay, so if light is coming from here and here, you must all have studied, so it is just like this. Okay, this light rays will goes like this, and it will form a image in the background, right? So here the image will be formed. So this is so the concave lens is diverging, and concave lens is diverging, and convex lens is your converging, right? So here I have mentioned few differences as well. So if you can see, after refraction, light rays meet at one point to appear to meet. Okay, in converging or convex, it's very simple. Okay, in convex lens, it is converging. See, if it is like this, so it is going to converge, right? It is converging and forming the image. However, in concave lens, it is diverging and it is forming the image at the background. Okay, so this is what the difference between concave and convex lens. So basically, diverging lens is your concave lens, because it is diverging the light and it is forming the image in the background, right? So this image is small object as compared to the real image. Okay, that is virtual image. Sorry, that is real image. Okay, the real image is always slow, always small, and virtual image is always large. Okay. Now the cylindrical lens bends the light rays to, see, and a spherical lens bends the rays to what? See, this is a spherical lens, right? This is a spherical lens. So this is a spherical lens. So spherical lens, you will find here it is a focal point. Okay, so spherical lens bend its light to a focal point. However, if it is a cylindrical lens, so cylindrical lens is like this. If it is a cylindrical lens, then it will be into the focal plane. Okay, so focal line. So this is the focal line. Sorry, this is, if light rays are coming like this, so it will converge into a focal line. It can converge into here, okay, any point, but it will be in a focal line. Okay. Now see, as we have discussed there, right, here in this structure, just a minute. So here we have seen the structure three-dimensional, right? Now if you can see this structure, okay, if you can see like this structure, here you can see, here is a space, right? Here is a space, and here is a space, right? Now see the structure very carefully. I am going to analyze this particular structure, this particular aspect. Now come to the two-dimensional picture. So see, if you have observed, again I will show you, if you are able to observe, just observe at 2 point. Okay, I'm just letting you know where you have to observe. You have to observe at this particular positions and this particular positions. Okay, so if you can just observe here, okay, so there is a space between cornea and lens and this entire chamber. Okay, so here I can say, tell you that this particular area is filled with a fluid. Okay, if you can see your anybody's eyes, you will find a glossy fillings, that is very what kind of view I can say, this is shiny fill, right? So that is basically because of what? That is basically because of this fluid, and that is called your aqueous humor. Okay, so the anterior chamber, that is, so the anterior chamber, just a minute. So the anterior chamber that is responsible, or that is present between the cornea and lens, this is your lens, okay, and this is your cornea. So this area is basically filled with your aqueous humor, and similarly the aqueous solution between lens and retina. So this is your lens, and this is your, this membrane is your retina, right? So this entire region is filled with your vitreous humor. Clear? Okay. Now come to this question, right? Now which of these has lowest refractive index? Okay, so here I have mentioned few refractive index, so that in of air it is 1. Okay, that is the lowest, and all these have more density, so that's they have more refractive index. So refractive index is directly proportional to the density of the molecule. More the density, more the refractive index. And we all know cornea, aqueous humor, vitreous humor are solutions. They have dense solution. Okay, so these all have highest refractive index, but air have very less refractive index. Now I have just put one image. So whenever light rays passes from lower density to higher density, the angle of incidence or angle of reflection is always less than the angle of incidence. Okay, but and vice versa, if it is traveling from denser medium to less denser medium, suppose I can say, suppose here it is traveling like this and traveling to here, then it will move like this, so that this angle is less and this angle will be more. Okay, so this is the law, that Snell's law rather, this is the law that whenever light is passing from lighter medium to the more denser medium, the angle of reflection will be less in denser medium. Okay, so denser medium में हमेशा angles कम रहेगा, जबकि lighter medium में it will diverge always. Okay, now for refractive power of the lens increased by the degree of 15 diopter, the sap of the lens क्या होगा? Okay, so if you can see here, two type of, I have given two examples, right? So this is your convex lens, right? So if we are considering the convex lens, so see here, this is your focal point, here this is your focal point. Okay, so more the radius or more convex it is, less the focal point, less convex it is, more the focal point. Okay, that is your focal length, right? So this distance is more in case of less convex lens, right? Now power is equal to 1 by f. 1 by f means 1 by, it is reciprocal of your focal length. Okay, so that means that less the focal length, more will be the power. Okay, that means more convex the lens is, more the power is. Okay, so that's why the shape of the lens needed to be more convex. Clear? Okay, now what is ametropia? Okay, so I am going to discuss here in this slide about the diseases, what is various abnormalities that we basically found. Okay, so ametropia is basically your normal vision. Okay, you are here, you can see the light rays is coming and it is completely focusing at the point that is called your retina. Okay, so this is your structure. This is the what we are looking for. This is what a proper normal vision is. Now myopia क्या होता है? What is myopia? It is the near sightedness. Okay, so when a objective is near, you can see it properly. Okay, but when a objective is at distance, you will not being able to see it properly. Okay, so what is the problem is here? Here you can see, here it is focusing before the retina, here it is retina, here it is focusing, right? So that's why the image is forming in front of the retina. Okay, if it is forming in front of the retina, our nerves is not being able to see that whether there is a signal or not. We are not being able to form a proper image, right? So for that, what we do, we use concave lens. So what concave lens do? It diverges the light, right? So it will see, if you can see here, it is diverging the lens a bit, line a bit, and further the lens is converging so that it is going to focus at the retina. Okay, so what it is important to remember is that myopia is your short sightedness. That means short sightedness means short can be visible, but image at distance is not able to visible. Okay, and to solve that, we need concave lens, biconcave lens. Okay, similarly hyperopia क्या होता है? What is hyperopia? Far sightedness. Okay, that means we can see the object that is very far, but we are not being able to see the object that is near to us. Okay, why? Because it is focusing below, after the retina. Okay, it is focusing after the retina, you can see like this. So image is forming behind the retina. So in order to make it to form in the retina, we use by convex lens. Okay, so that it will converge, so that it will form in front of the, it will form in more front, like, because it is forming at the back of the retina, right? So in order to make it to form in the retina, we use convex lens. So this is what you need to important: normal vision, ametropia, myopia, short sightedness, hyperopia, far sightedness. Short sightedness में क्या होगा? Near you are being able to visualize nearer object, but you are not being able to see distant object, and that's why to solve that we need concave lens. Okay, you can just imagine, right? Concave lens, we have already discussed, concave will be diverge the lines, right? If it will diverge, then our lens is there to converge, right? So lens will converge and it will make it form in the retina. Similarly, in hyperopia क्या होता है? The image is forming after the retina, right? That's why we need to have some kind of correction that will form in front of the retina, and for that we need to converge the light, and to converge we can use convex lens. Okay, so that it can be formed in the retina. Clear? Okay, now the question 8 and 9 is that rhodopsin consists of, here you can see rhodopsin precursor are transretinal and all transretinal. Retinal is basically vitamin A. Retinine is basically better known as retinal. Okay, retinal is a aldehyde, and it is basically, you can see in this pathway that rhodopsin in presence of light, it converted to beta-rhodopsin, then luminorhodopsin, methylrhodopsin, and all the transretinal and cisretinal, right? So this transretinal is basically vitamin A, that further converted to cisretinal and to rhodopsin, active rhodopsin, that plays a major role in the rod cells. Okay, and that rod cells are basically helpful to visualize the signals at low light condition, dim light condition. Clear? So rhodopsin basically consists of photopsin and retinine. Okay, now retinine is also known as retinal, and retinal is basically a vitamin A precursor. Clear? Okay, guys. Now the next question is that in which condition CGMP pause opening of sodium channels in rod cells. Okay, so we can see here that rods are exquisitely sensitive to light and can even detect single photons, right? So rods have the ability to visualize the very dim light. They allow vision in very dim light. Okay, so they are exquisitely sensitive to light and can even detect very single photons. सपोज लो लाइट में कैसे देख सकते हैं हम लोग, अगर थोड़ा सा भी सिग्नल्स है if rods can detect, then only will be able to define that, right? So for that, rods in dark condition, what they do, okay, there are high concentration of CGMP in our body. Okay, so you can see in dark क्या होता है, dark जब होता है so there will be no signal, that is the point. So we don't have signals, right? So we need suppose neurotransmitters get released, because there is very very few signals, right? Basically these are neurotransmitter is because that there is postsynaptic neurons is inhibited, right? Because there is no signal, right? Now neurotransmitter suppose release हुआ because there is some kind of signals, okay, and you want to visualize. Now in dark what will happen, the CGMP condition is CGMP is more present in dark in our body. Okay, so CGMP bound to the sodium channels, and the sodium channels is okay, the sodium channel is basically responsible for depolarization. Okay, so the cells depolarized release inhibitory transmitter prevent other neurons firing. Okay, so the cell will depolarized, okay, because it is dark and we have sodium channel open because of the CGMP. Okay, so cell got depolarized. Okay, and this depolarization will release the inhibitory transmitter. So this is a signal to inhibit the secretion of neurotransmitter. Okay, so that the rhodopsin are inactive and the sodium channels get opened. Okay, so the rod cells are depolarized. Okay, so the rod cells are depolarized so that they can perform the function. Okay, in the presence of light, the cis-retinal to trans-retinal conversion and further G protein transduction CGMP, it leaves sodium channel. So now again sodium channel will stop, closes because of the CGMP level get decreases. Now rhodopsin get active, okay, and rod cells will be hyperpolarized. Okay, so rods are exquisitely sensitive to the low light. Even there is low light, even it can sense. Okay, and the formation or the activation is basically done in presence of light, that so that it can even act in the dark. Okay, so even in the dark it has the ability to define that very dim light. However, cones are only capable to sensitize light in higher concentration. So they are responsible for colors, they are responsible for bright lights, but for dim lights it is rhodopsin that is responsible for the vision. Okay, so whenever there is a small amount of light, so rhodopsin and further G protein transduction and CGMP phosphodiesterase and CGMP will active the sodium channels, so that CGMP get its sodium channels. Okay, so these all things are happen in the dark. So even in the dark this all things can happen. Even a small amount of light can activate rhodopsin. Okay, so the rhodopsin is very sensitive to the light. Even a small amount of light is able to responsible for the conversion of cis-retinal to trans-retinal, and the CGMP concentration will decrease, and that will activate the rhodopsin for sensitizing the signal. Okay, now this is what about rod cells with polarization and formation of signals so that light can transmit, or we can sense the visualization in the dim light. Okay, so this condition is that in dark condition CGMP is concentration is high and it opens the sodium channel. okay, and sodium channel opening will inactivate the rhodopsin. However, a small amount of light even can activate the rhodopsin. Okay, clear? So this is what about today, and we'll discuss further on the next Friday. Till then, have a good day and goodbye. And if you have any doubt, you can just come to discuss. Okay, guys, then see you. Thank you. Bye bye.